Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning session in Biomolecules. Today in this session we learn about the different protein configurations. For convenience, to explain the complicated structure, biochemists have described 4 different configurations for protein structure. They are primary structure, secondary structure, tertiary structure, and quaternary structure. In mathematical term, they can be represented as primary structure as like this, then secondary structure like this, then tertiary structure and quaternary structure will be represented like this respectively. The first 3 organizations, that is primary, secondary and tertiary, can exist in a protein containing single polypeptide chain. That is primary, secondary and tertiary. You can see these 3 structures in a protein containing single polypeptide chain. But the quaternary structure exists only when protein has more than one polypeptide chain. The primary structure of protein consists of linear chain of amino acid. It is just the linear chain of amino acids. They are joined together by peptide linkage and is unfolded simple structure. This linear unfolded structure assumes a helical shape to produce the secondary structure. So primary structure folds upon itself to form the helical structure, that is called the secondary structure. This in turn may fold in certain specific pattern to produce twisted 3-dimensional structure. Twisted 3-dimensional structure upon further folding, that is called tertiary structure. Some proteins are made up of subunits of similar or dissimilar type of polypeptide chain. They are made up of same type of polypeptides or different type of polypeptides in the form of subunits. These subunits interact in specific manner with each other to give rise to quaternary structure of proteins. So there are 4 configurations: primary, secondary, tertiary and quaternary. Now we will see each structure in detail. Primary structure refers to the number of constituent amino acid. Primary structure of the protein is the sequence of amino acid in the chain. It refers to the number of constituent amino acids of a polypeptide chain. These amino acids are stabilized or held together by peptide bond between the alpha carboxyl group of one amino acid and NH2 group of another amino acid. So that specialized CONH linkage links the 2 adjacent amino acid. It determines the eventual shape of the protein and hence its function. This type can have one or more peptide chains. Next we will move on to the secondary structure of protein. This linear chain of amino acid in the primary structure again folds upon themselves to form globular structure due to the presence of hydrogen bond. Thus, folding and hydrogen bonding between the neighboring amino acids results in the formation of rigid tubular structure called the helix. This forms the secondary structure of the proteins. This secondary structure refers to the steric or spatial relationship of amino acids that are near to each other in the amino acid sequence, in the string of amino acid. Based on the nature of the hydrogen bonding, Pauling and Corey identified 2 types of the secondary structure. One is alpha helix and another one is beta pleated sheet. First we will take up alpha helix. Pauling and Corey found that polypeptide chain with a planar peptide bonds would form a right-handed helical structure by simple twist about the alpha carbon to nitrogen and alpha carbon to carboxyl carbon bonds. So they are called as the alpha helix. So these were termed by Pauling and Corey as the alpha helix, and is called so because of the mobility of the alpha carbon atom. Alpha helix is a rod-like structure that is tightly coiled polypeptide main chain forms the inner part of the rod. Next, coming to the beta pleated sheet, this is called so because Pauling and Corey elucidated the secondary structure. The first was alpha helix. Next, to differentiate it from the alpha helix, it was called as beta sheet. The formation of the beta sheet depends on the intermolecular hydrogen bonding through, that is, in addition to the intramolecular hydrogen bonds are present. That means you have intermolecular hydrogen bond and intramolecular hydrogen bond. The pleated sheet structure is formed by parallel alignment of number of polypeptide chains in a plane with hydrogen bonds between the CO and NH groups of the adjacent chains. The R groups of the constituent amino acid in one polypeptide chain alternatively project above and below the polypeptide chain. So they form the hydrogen bond between the polypeptide, leading to the two residues or the two side chains of the adjacent polypeptide chain. They project and they result in the formation of the hydrogen bond. These beta sheets are common and are rich in glycine, alanine and serine. These are rich in glycine, alanine and serine. Silk, the synthetic fibers like nylon and other synthetic fibers, they have the beta pleated sheet. This beta pleated sheet differs markedly from rod-like helical alpha helical structure. So now see the difference between the alpha helix and the beta pleated sheet. If you take the alpha helix, it's a rod-like structure, helical structure, coiled polypeptide chain arranged in spiral structure. It's a sheet-like structure. It's not helical. It's a sheet-like structure composed of two or more polypeptide chains. In case of alpha helix, all the peptide bond components participate in the hydrogen bonding. Here also all the peptide bond components participate in the hydrogen bonding. In case of alpha helix, all the hydrogen bond is intra-chain, that is within the polypeptide chain, and it is abundant in hemoglobin and myoglobin. Whereas in case of beta pleated sheet, interchain between the separate polypeptide chain. You'll find interchain hydrogen bond along with the intra-chain in single polypeptide chain folding. So you'll find both inter and intra-chain hydrogen bonds in beta pleated sheet. In case of alpha helix, the spiral of alpha helix prevents the chain from being fully extended. But in case of the beta pleated sheet, the chain are almost fully extended and relatively flat. They may be parallel or anti-parallel. If the protein is made up of more than one polypeptide chain, if the two polypeptide chain run in the same direction, they are called parallel polypeptide chains, and the beta pleated sheets between these two polypeptides are called parallel beta pleated sheet. If you find the beta pleated sheet between the two polypeptide chains running in opposite direction, then they are called as anti-parallel beta pleated sheet. So this beta pleated sheet usually has the extended conformation compared to the alpha helix. If you just consider the axial distance between the adjacent amino acid in the beta pleated sheet is 3.5 angstrom, whereas in case of the alpha helix it is 1.5 angstrom. So this is the reason why we have the extended structure in case of the beta pleated sheet. It's around 3.5 angstrom unit in case of the beta pleated sheet. Next, we'll move on to the tertiary structure of protein. The tertiary structure defines the specific overall 3D shape of the protein. The tertiary structure is based on various types of interactions between the side chains of the peptide or the polypeptide chain. This tertiary structure involves folding of the helices of the globular proteins. It refers to the spatial arrangement of amino acid that are far apart from one another in the linear sequence, and to the pattern of disulfide bonds. The tertiary structure formed by folding of the chain or overall folding. The X-ray crystallographic studies have revealed the detailed 3D structure of more than 300 proteins, and it is stabilized by the non-covalent interactions like hydrophobic interaction, hydrogen bond, ionic bond, and it is also stabilized by the covalent interactions like disulfide bond and the primary bond, that is the peptide bond. The best example for the tertiary structure is the structure of the myoglobin. So myoglobin was the first protein, the complete tertiary structure was determined by X-ray crystallography. It has 8 alpha helical region and it has no beta pleated sheet. Hydrogen bonding stabilizes the alpha helical region. It consists of the single polypeptide chain of around 153 amino acid residue and include the prosthetic group, that is the heme group, that will give the red color to the muscle cell as it is rich in the muscle cell. So this myoglobin stores the oxygen as a reserve against oxygen deprivation. So this myoglobin is relatively small oxygen binding heme protein found in muscle cells. It's first globular protein to have 3D structure, and it is around 153 amino acid residue, and it has the prosthetic group, that is the heme group, and its heme group is identical to hemoglobin heme group. It is present in the muscles of dividing mammals such as the whales, seal and porpoise. The heme group is in case of myoglobin is responsible for a deep red brown color of the muscle. The function of myoglobin is to bind to the oxygen in the muscles and to enhance its transport to the mitochondria, which consumes oxygen during respiration. So that is regarding the tertiary structure. Next we will move on to the quaternary structure. The biological function of some biomolecules is determined by multiple polypeptide chains, that is multimeric protein. The arrangement of the polypeptide or in the form of subunit is called quaternary structure. Subunits are held together by non-covalent interaction. The best example is hemoglobin, which has the subunit composition of A2, B2. So globular protein consists usually of quaternary structure and they are made up of 2 or more polypeptide chain. Each polypeptide chain in such protein is considered as subunit. So these chains may be identical or different in their primary structure. So this specific association of number of subunits into complex large sized molecule is referred as quaternary structure. In other words, quaternary structure refers to the spatial arrangement of subunits. The same forces like disulfide, hydrogen, hydrophobic, ionic bond stabilize the protein structure. The best example, as I told earlier, is hemoglobin. Heme means blood, globin means protein. It's an oxygen transporter in erythrocytes, constitutes about 90% of the proteins of the red blood cells. It is tetrameric protein. It is having 4 polypeptide chains. The 4 chains are held together by non-covalent interactions. Each chain contain a heme group. Each chain contain a heme group and a single oxygen binding site. There are 2 types of hemoglobin, HbA and HbA2. HbA, that is hemoglobin A, principal hemoglobin in adults, consists of 2 alpha chains and 2 beta chains. The hemoglobin A2, adults also have minor hemoglobin, that is around 2% of the total hemoglobin, called hemoglobin A2, which contains 2 delta chains in place of beta chains. HbA and HbA2 are the postnatal forms of hemoglobin. So this hemoglobin weighs around 64.5 kDa, provides an example of unidentical protein subunit interaction, since it is made up of alpha and beta chains, 2 alpha and 2 beta chains. Since hemoglobin is about 4 times larger than myoglobin, so little more effort was required to elucidate the 3D structure of the hemoglobin compared to the myoglobin. John C. Kendrew elucidated the 3D structure of horse hemoglobin. X-ray analysis revealed that hemoglobin molecule is nearly spherical with a diameter of 55 angstrom. Hemoglobin protein consists of 4 polypeptide, 2 alpha and 2 beta. The polypeptide portion is collectively called as globin. Alpha chain has valine at N terminal and arginine at C terminal, whereas beta chain has valine at N terminal and histidine at C terminal in case of beta polypeptide chain. Each alpha chain is in contact with beta chain. In contrast, there are few interactions between 2 alpha chains or between 2 beta chains. Each chain has a heme prosthetic group in a crevices near the exterior of the molecule. Heme group is involved in binding of the oxygen. If you just see the amino acid composition, there are around 141 amino acid in the alpha chain and 146 amino acid residue in beta chain. The protein has around total of around 574 amino acid residue. Each of the 4 polypeptide chain has a characteristic tertiary structure and is stabilized by non-covalent interactions like hydrogen ionic hydrophobic interaction and covalent interactions like disulfide and peptide bond. Thus the 4 polypeptide chains fit together almost tetrahedrally to produce quaternary structure. So this is regarding the classification of protein based on their configuration like primary, secondary and tertiary. So in this session we learned the different structural organization with few examples refers to the quaternary and tertiary structure. I hope you understood this session. Thank you.